Hello all. In this next video lecture, we're going to begin moving into Chapter 6 to start off Unit 2. And yes, I know we're bypassing Chapter 5 for now as part of Unit 2. We'll actually come back and pick that one up at the end of Unit 2. And the reason for that is Chapter 5 covers viruses, and I feel very strongly that you need to have some background in microbial genetics before you do that virus chapter. I think the virus chapter will make a lot more sense once we've been through Chapter 8 on genetics. So Unit 2 is a fairly long unit, kind of like Unit 1, and I would say that this unit gets into more nitty-gritty kind of details than Unit 1 did. So just a heads up on that. Unit 2 will require quite a bit of study time. All right, and so to start off with Chapter 6, we're going to be looking at microbial nutrition and growth, and we're mainly going to be focusing in on bacteria. So if we think about the two groups of microorganisms that are responsible for the most human disease, at least in the United States, bacteria and viruses. Well, viruses don't metabolize. Viruses depend on a host to reproduce them. So they are not capable by themselves of metabolizing and growing. So when we're thinking about microbial nutrition and growth, we've got to think about living microorganisms. Now, some things in here are going to apply across all living things, not just microorganisms. And like I said, we'll really kind of mostly be homing in on bacteria as the group that we're studying primarily here in Chapter 6. All right, so kind of to start off with, Microbial Nutrition Part 1. And what do we mean? So we're thinking about microorganisms, and what do we mean by microbial growth? So as you guys will see early on in this chapter, it's not so much we're thinking about an individual bacterial cell or an individual protozoan or an individual yeast cell growing and expanding in size. Instead, when we refer to microbial growth, we're really thinking more about an increase in population of the microbes. So think about your lab simulations that you did where you were culturing and growing microorganisms in your virtual laboratory. Growth meant the numbers of those organisms were increasing. All right, so for example, here's a petri dish with some sort of culture medium in it, an agar medium, and you've got colonies all over that plate. Well, each of those colonies started from a single bacterial cell that divided over and over again to create those colonies. So we say the colonies grew on the plates. Or if you had a liquid broth culture in the laboratory, remember turbidity, that term which refers to cloudiness of a bacterial liquid culture. And so if a culture turns turbid, you know that there are a whole bunch of microbial cells in there that have increased in population and have caused that culture to turn cloudy. And this kind of gross picture over here, how do we know microbes are growing, pathogens are growing in the human body? So if you have something like this on your skin, this lesion, and by the way, this would be called an abscess because we see pus in there. And when you see pus on a wound like this, or even inside the body, all that yellowy, nasty looking liquid there includes fluid that has moved out of the blood vessels and into the tissues. That's what causes the swelling associated with inflammation. But then most of that yellow color is coming from white blood cells that have exited your blood vessels. We'll learn more about those later in the semester. And they have migrated into the tissues to try to fight the infection. And so you see an accumulation of all those white blood cells. They die. They don't live forever. They come in and swarm in and fight off the bacteria and die there, and they accumulate, and that leads to pus. And generally, when you see pus, that's a strong sign of a bacterial infection as opposed to an infection with some other type of microbe in those tissues. All right, so in order for you to be harmed in your body by a pathogenic microbe, it has to be growing. It has to be increasing in numbers. So if you just have 20 of some sort of pathogenic microorganism living in a tissue lesion somewhere, that's not going to make you sick. You're not going to be aware of it. It has to increase in numbers. It has to grow in order to actually start to harm you and cause you problems. All right, so what drives growth? Metabolism, that term refers to the sum of all the chemical reactions that take place in a cell or an organism. And then, of course, reproduction. So we're thinking about little single-celled microbes. They have to divide in order to increase in numbers. If these things are happening, then you're going to have growth of these microorganisms. All right, so when you guys take anatomy and physiology, you learn about human metabolism and growth. And as we're going to see, microorganisms, as we already know, are very diverse. So there are microorganisms that have adapted to almost every possible environment on the planet Earth. So what that means is with microorganisms, their metabolisms are not all going to be identical. They are going to differ according to what that particular organism's needs are. What are its adaptations going to be? Metabolism. So all these chemical reactions that take place, some of those are required to make energy available for growth. And if we think about, things that living organisms do that non-living things don't do, one is that they harness energy from the environment. It takes a lot of energy to make individual cells be able to operate and survive and reproduce all their parts and stay healthy and functioning. It takes a lot of energy. So you've got to be able to absorb this energy in from your environment in some way. And then also, as we'll see, living things have to be able to assimilate chemical compounds and use or generate the ones that they need to construct all their cell parts and their cell machinery in order to be able to survive. All right, so all this metabolism is critical for survival. All organisms have metabolic needs of some sort that have to be met. And if those needs are met, then you're going to grow. You're going to be able to reproduce and survive. And that's true for any living thing, not just microorganisms. All right, so when we're thinking about all living things require nutrients, okay? Well, we've all heard of nutrients before. Obviously, we need nutrients in order to be able to survive, and that's true for any living thing. And nutrients can be classified as macronutrients. Those are things that are needed in large quantities for the cells of an organism to be able to function. And then there are others that are called micronutrients, and another term for micronutrients is trace elements. So these are things that you need in tiny amounts, and those are generally mineral ions. So if you are taking a vitamin supplement at home as part of your daily routine, you look on the back and you see a listing of things like copper and zinc and manganese and things like that. Those are all trace elements that you need in small quantities in order to be able to survive. Generally, they're important for one or more of your enzymes to function properly. And the same is true for all living things, including little lowly microorganisms. They have micronutrients as well as macronutrients that they need to be able to survive. So some key macronutrients are very familiar organic macromolecules that you hear about in every biology class: carbohydrates, lipids, proteins, and nucleic acids. And then we've learned these building blocks before. And then if you take the building blocks, sugars or monosaccharides of carbohydrates, and break those down into their individual elements, sugars are generally built from carbon and hydrogen and oxygen atoms put together in those particular patterns to make them sugars. Lipids are generally also built from carbons and hydrogens and oxygens, and sometimes phosphorus in the phospholipids that you find in your cell membranes. It's just that these lipids are put together in different patterns compared to sugars, and that's what makes them lipids instead of sugars. Proteins are built from amino acids. If you break amino acids apart into their individual atoms, those are mainly going to be carbon, hydrogen, oxygen, and nitrogen with some sulfur, some phosphorus. And your nucleic acids, the building blocks of those are nucleotides. You break those apart, carbon, hydrogen, oxygen, nitrogen, and phosphorus atoms. So you can probably see here these organic molecules are primarily built from just a small subset of all the elements that exist out in the universe. And of course the main one is carbon. All of these have a carbon backbone as part of their molecular skeletons. Lots of hydrogen, lots of oxygen in there as well, just not as much as you have with carbon. Nitrogen, sulfur, and phosphorus. So sometimes those elements, those are referred to as the key macronutrients, the main elements that living things need. There's kind of an acronym for that too, which is, I've seen it as CHNOPS, CHNOPS, as a way to remember what those major elements are that living things need, or CHOMPS, CHOMPS. Organic versus inorganic, we've discussed that a little bit before back in the chemistry section in chapter 1. Remember, organic molecules are molecules that have a carbon backbone, and were traditionally associated with living matter rather than non-living matter. All right, so here's a table from your textbook which kind of emphasizes these things a little bit further. If you looked at the chemical composition of a single Escherichia coli cell, what's in there? We're thinking about the dry weight here, so we're removing the water, so no H2O in there. The dry weight, what's left over? Organic compounds make up by far most of the dry weight of a bacterial cell, and it'd be the same for our cells as well. And notice that 50% of that dry weight comes from proteins. So in your biology classes, you learn that cells manufacture lots of different kinds of proteins. They have lots of different jobs that they need to perform. They're very diverse. They're sometimes called the chemical workhorses of cells. So the fact that 50% of the dry weight of a cell is consists of different proteins just emphasizes that for us. Nucleic acids, RNA, about 20%, DNA, 3%. So we hear about DNA all the time, so you would think that might be a greater contributor to the dry weight of a cell, but it's actually only 3% in E. coli. And remember, your DNA is really like a set of recipe books, genetic recipe books, that provides instructions in a molecular type chemical language that allows the cell to build all of its parts and operate. So it turns out that not a lot of the dry weight actually is devoted to those blueprints. The blueprints, though, instruct the cell to build all of these amazing parts and so forth that are needed for it to be able to function. And you have your carbohydrates and your lipids are about 10% apiece. Then other inorganic compounds, still very important, but they only make up about 3% of the dry weight of a cell. Then over here, if you break all of these things down, chop them up into individual atoms, elements, notice carbon, 50% of the dry weight of a cell. So again, that emphasizes that we are carbon-based. Living things are carbon-based. Oxygen, 20%. Hydrogen, 14%. Then you get down here into some things like potassium and sodium and calcium, and you hear about those a lot when you take your A&P classes, those different ions in our body fluids that play such important roles in the muscular system and the nervous system. But notice they're only like 1% or even less of the dry weight of a cell, so they're able to carry out all those important functions that they have in cells, even though they don't contribute much to the dry weight. All right, the next topic. All living organisms, and again, we're not including viruses here, we're thinking about truly living things. They need what we call the carbon sources for their survival. So we just said living things are carbon-based organisms, so you have got to take in carbon-based nutrients in order to be able to survive. And the carbon-based nutrients that an organism uses are referred to as its carbon sources. And all living things have to have a source of energy. Okay, so living things can be classified according to where they obtain their carbon. So if your carbon source, your carbon-containing nutrient, is the carbon dioxide in the atmosphere, you're classified as an autotroph. And the reason for that is because you are able to take carbon dioxide, and those carbons in there, you're going to perform as part of your metabolism lots and lots of chemical reactions that are going to use the carbons here and build all of these important molecules that you see all over here. All of these are carbon-based, or proteins, or nucleic acids, etc. So if you can do that, you are an autotroph. Troph in biology generally refers to feeding. Auto means self. You're a feeder in the sense that all living things need these carbon-containing compounds, but you can build your own, starting with CO2 from the atmosphere. Now, organisms like us and the vast majority of the microorganisms we study in this class are called heterotrophs, which means other feeders. So that means you're going to obtain carbon-based organic compounds from your surroundings. You've got to take in organic molecules that have already been created by other organisms. Now, that doesn't mean you can't build some of your own organic molecules, but you can't build them all. You've got to bring in some building blocks to make a contribution. So that's carbon source, autotrophs and heterotrophs. And then energy sources, where are you getting your energy from? What's the source of energy? All living things have to harness energy from their surroundings. If you get your energy from sunlight, you're called a phototroph. So organisms that do photosynthesis, for example, are phototrophs. And if you get your energy from chemical nutrients, like we do, and also the microorganisms that we're primarily concerned with in this class, you're a chemotroph. Then let's see how we can put these together. So you can put those two things together and categorize living things into several different categories. 4 main ones that we have here. Okay, so autotroph and heterotroph, again, that refers to where are you getting your carbon from? What's your carbon source? And then photo and chemo refers to where are you getting your energy from? Is it coming from sunlight, or is it coming from chemical nutrients that you break down? You break bonds in those chemical molecules, and that releases energy that you can use to do work. All right, then you can combine those things. So we have the photoautotrophs. They get their energy from sunlight. They get their carbon from carbon dioxide in the atmosphere. And those are pretty common. Algae and plants, there are a number of bacteria that do that as well. Then you have chemoautotrophs. So they're autotrophs, so they get their carbon from CO2, and they're able to change that into sugars and amino acids and so forth and so on. But instead of getting their energy from sunlight, they harness their energy from simple inorganic molecules. So they have kind of unusual lifestyles. And we don't study those really in this class all that much. Now the heterotrophs over here, photoheterotroph, you get your energy from the sun. And your carbon, though, rather than coming from CO2, you use organic molecules that have been made by other organisms. And finally, we have our chemoheterotrophs, and these are the organisms we're primarily concerned with in this class. And we fall into this category as well. And so here you are getting your energy and your carbon from chemical nutrients, organic nutrients. All right, so obviously we get our energy and carbon sources from the foods that we take in. The foods that we eat are all organic molecules, I hope. I guess if you're eating Twinkies or whatever, there are probably some chemicals in that white stuff that are not in the organic category. And then the microorganisms that we study in this class feed off of us when they enter the body. So they're breaking down our tissues and our organic molecules and using those as energy and carbon sources. So they fall into the chemoheterotroph category as well. There are a couple of special examples of chemoheterotrophs. Saprobes are decomposers, so they strictly feed off of the organic matter of dead organisms. So any organism that functions as a decomposer out in the environment falls into that category. Fungi, lots of bacteria do that as well. Then parasites, we've talked about parasites before. They feed off of the living tissues of a host. So obviously we're concerned with, and we tend to use that word parasite, you tend to think of like a parasitic worm, but that term can also be used to describe any organism that lives in a host and harms the host as part of that process. Okay, pathogens There are some pathogens that are saprobes out in the environment. There's some fungal pathogens that live as saprobes in the environment, but if they get into the human body, they become parasites. And we heard a little bit about the dimorphic fungi before. Remember those from back in Chapter 4? There are some types of fungi that live as molds in the environment, but when they enter the body, they convert to a single-celled yeast form, and they can cause some very severe respiratory infections. And an example of that is a fungus called histoplasma, which causes a disease known as histoplasmosis, and there are some other ones that are somewhat similar. But histoplasma is the one that you would most likely encounter in this part of the United States. So histoplasma is a mold that grows in bird or bat droppings, so it's a decomposer. It breaks down the organic molecules present in those substances. But if you inhale this, so these are hyphae, remember those, chains of fungal cells, and these are spore-producing reproductive structures here. If you inhale those spores, like this poor little guy over here, they go down into the lungs, and then this histoplasma fungus grows as a yeast, and eventually the yeast get carried through the bloodstream to the skin, and you can develop skin lesions from it. So it's a pretty nasty type of infection. The disease is histoplasmosis, a little bit more of a problem north of here up toward the Ohio River, Louisville, Cincinnati, that area, but there can be some cases in this part of the country as well. But that's a dimorphic fungus. It's living as a saprobe out in the environment. It becomes a parasite once it enters the human body. All right, so bacterial culture media. So we've learned about these before, but now that we're thinking more about growth in detail, those culture media that we learned about in Chapter 2 and also in your lab simulations, they have to provide all of the macro and the nutrients that are needed for the growth of a microorganism. You've got to supply all of those things in order for the microorganism to be able to grow artificially in the laboratory in this culture medium. So not just macronutrients, you've got to provide any micronutrients that are required. Growth factors, we'll talk a little bit about growth factors, and you might be thinking, what's the difference? A growth factor is generally a larger organic molecule that's not a protein, not a sugar, not a fatty acid that an organism needs to be able to survive. So it's something the organism can't make on its own, but it needs it for survival. Like a lot of vitamins would fall into that category. Vitamins are organic molecules, carbon-based, but they don't really fall neatly into the protein or nucleic acid or a carbohydrate or lipid category. So different organisms have different growth factors that they need to be able to survive. There's another term you should be familiar with too, and that is fastidious. And so what are fastidious organisms? So based on the name, you might think a fastidious organism is one that would grow fast in the laboratory. That's not the case. This actually refers to a picky organism. So it's one that needs more growth factors in its culture medium than you would typically find in kind of a standard nutrient agar mix that you would prepare in the laboratory. A good example of a fastidious organism, Haemophilus influenzae or influenzae, and now that's a bacterial organism. It's not a virus. I know it sounds like the name of the influenza virus that causes the flu, and it does cause a respiratory infection that could mimic the flu, but it's actually a bacterial organism, and if it leaves the respiratory tract, gets in the bloodstream, and goes to the brain, it can infect the meninges of the brain and can cause meningitis, especially in kids. So little kids get vaccinated against this organism to try to prevent that illness. But anyway, it's a fastidious organism, so it requires lots of growth factors in its culture medium in order to grow under laboratory conditions. Remember, in a laboratory, traditionally in a microbiology laboratory, if we wanted to identify, do research on, characterize, run tests on a microorganism, we needed to be able to grow it in the laboratory. And some of that is changing as our technology is improving. We can do a lot now if we just have the DNA of the organism. But still, we're not quite to the point where that's fully sufficient. So we still need to be able to grow these organisms from low numbers to higher numbers to have enough of them to be able to run tests on them and identify them and characterize them and do research on them if you're working in a research lab. So you need to know what those fastidious organisms require in order to survive. And you guys will see, and if you're around clinical microbiology laboratories, it's very common for microorganisms from patient samples to be grown on something called blood agar. So it's a standard agar medium that has lots of nutrients in it, but they add sheep's blood to it because sheep's blood is so rich in growth factors that a number of pathogens will grow better on the blood agar as compared to some other standard types of culture media that are used in microbiology labs. Okay, so that was kind of an introduction to microbial nutrition, and we will move on. I'll stop this video lecture here, and we'll pick up and continue on the next video lecture.